it's a bit strange to talk about social interactions where basically we're doing the same session, but without the social interactions between us. But still, it's a very nice idea to try to get it all online. So thank you very much for doing this. So I'll dive right in. I slightly changed the title, but it's the same talk, of course. And basically, I'm going to talk about emergent structures as a result of interactions between growth-driven systems. And I'll explain what I mean. So maybe a little bit about my lab. Basically, in the lab, we study the physics of growing systems and plant behavior specifically. And when I say plant behavior, people usually kind of raise their eyebrows because we're used to seeing a plant, such as what you see here in the on the screen, right? Like the plants in our office or at home, and they're generally static. They don't move, and they definitely don't look like they're behaving at all, right? However, if we go to YouTube and we look for time lapses, we'll see that there are many videos where we see the plants moving a lot. Okay, so the point is basically that this is a problem of our own perception, because plants move a lot just by growth, and this is a very slow time scale, and therefore we're not able to grasp their movement. But as you can see here, for example, in this movie from BBC Planet Earth, where you see these twiners, climbing plants, searching for, right, they're in search for light, they're searching for supports, searching, you see on the right here, there's this one climber that's turning frantically until it finally, whoop, finds the support and then continues to grow. So when you watch these kind of videos, you might understand where behavior comes from, right? Where you can imagine that these plants are behaving. They have a strategy for growth, they make decisions, they have memory, and so on. One interesting thing about plants, as opposed to maybe other more commonly studied animals in the context of behavior, is that since the movement that you see here is due to growth, okay, so it's not self-propulsion, as in fish or worms or birds or whatever. And this means that since usually behavior is represented in motion, in plants we cannot separate between development or growth and behavior. So in order to understand behavior, we must have a good model for growth. And also in order to understand the dynamics of growth in plants, we must understand their behavior. So it's an intertwined problem. And the problem that I want to talk about today specifically has to do with interesting structures that are quite ubiquitous in biology. And here I'm giving just two specific examples, but there are many others, where basically you have a system that's made up of, or a structure made up of many individuals, where each individual is a growing system. So on the left, you have an example of a root structure. This is an X-ray or a CT of two root systems. And on the right, you have a portion of the brain hippocampus. Now, what you can appreciate here is that the structure, you can especially see it in the hippocampus, since you have different layers there, and you can see that the different layers have different structures, right? In one part it might look more blobby or random, and on the right it might look more polarized or all neurons in the same direction. And we know that these different parts have different functions. And we know that there is, of course, a connection between form and function. So we can imagine that the computational aspect of the brain dictates which kind of structure you need. The same goes for the roots. With the roots, you have a specific goal. The goal is to maximize nutrients and stay out of each other's way of the roots. And you'll see different root structures according to different environments that they have. And then the question is, so since these are all made up of many single individuals, many neurons or many roots, how do we get these structures? How do these structures emerge from these many individuals? So to do this, I want to zoom into the individuals. I call these individuals, basically, if they have one thing in common, is that they're all what I call a sensory growth system, all right? So they all move by growing. You have neurons on the top and fungi and plant shoots and, of course, plant roots. They all have senses for a variety of signals, chemical, light, gravity, and so on. But their movement is due to growth. Okay, so everything that you see here on the screen is all movement that is due to growth. Now, in the talk today, I will focus on plant tropisms, or the models that I will show here are inspired by plant tropisms. Tropisms are these growth-directed responses of plants to stimuli. But the results are completely general. They can be used for any other system as well, since, as you'll see, the models are not based on biological details. Now, one thing I want you to appreciate from the two movies that you see here at the bottom, on the left, we have an example of gravitropism. So the plants are put on the side, and the whole plant is full of sensors that tell them which way is down, the direction of gravity. And you see the shoots grow up. Now you'll see that they flip the pot, as they did right now. And you see that the plants they notice that the direction of gravity has changed, and they start growing up. But what you have is then, and the same goes with light, they grow to the left, and then the direction of light is switched to the right. And the final form of the sort of U-shape or curved shape here is that the structure of the plants basically encodes with it information of all the history. Okay, and this is an important thing to note. Can you still hear me over there?
So you're a little bit cut off.
Hello?
But now I think it's a little bit better. She did it. Is she not?
Everybody else is still there, right? Raise your hand if you can hear us.
Still here. How do you raise hand on the, where's the function?
Oh, it's on the participant list. You have the option, there's a...
Oh, cool, thanks.
Yeah, if you press participant.
If you go there, right.
Where's participant? Oh, in the bottom.
Right next to the share on my screen.
Oh, raise hand, okay.
Yasmine?
Oh, she's muted.
Hello?
Oh, yeah, now we can hear you.
Okay, yeah, it's only disconnected and now connected again. Okay.
Let's go with it. It's okay.
Okay, so is this okay if I start here, or did you miss more than this?
Maybe one slide
back, please.
Okay, so I was here.
Yeah.
And yeah, okay, and I was just saying that basically the final form of the plant, or whichever system that we're looking at, of these sensory growth systems, encodes within it basically the history of everything that the plant has ever sensed, right? As we saw in this U shape, it means that it first went to the left and then it went to the right. And basically, the nice thing about working on, I said that we're focusing on, I'll focus here on plant tropisms just because it's a good starting point. But we'll see that the models that I show here are not based on any biological details, and therefore are general and can be applied to other systems as well. So I want to continue now and present the mathematical model that we use in order to describe the dynamics of these plant tropisms. So very quickly here on the left, you see this is a wheat coloptile that was placed on the side, and it basically straightened up. It sensed the direction of gravity and it straightened up. So what we do is we parameterize this. We have a variable S, or parameter S, that goes along the center line of our shoot, of our organ, and it starts at S equals 0 at the base and S equals L at the tip. And now at every point S, I can define a local angle theta. Okay, so I have theta, the angle from the vertical, at some specific point S at time t. I can also, now that I have an angle, I can define a local curvature. The curvature is just the derivative of the angle. If the curvature is 0, it means that theta hasn't changed, it's straight, okay? And that's it. So now that we have the parameterization, we can also formulate an equation that describes the kinematics. So I'm showing here on the top 3 different plants with different scales, completely different species. On the left, we have wheat, which is relatively small. In the center, we have Arabidopsis inflorescens, which is larger. And you will see that if you saw there were many overshoots, the dynamics are different. And then on the right, we have poplar trees. These are all gravitropic experiments. And all of these can be explained with the equation that you see here below. So this is due to a beautiful work by Renaud Bastien et al. And company from 2013. So what you see on the left, we have basically the derivative in time of the curvature. So this is the growth response, the dynamics. And these are influenced by what you have on the right-hand side, which is the effective stimulus. So theta p is the angle of the stimulus, all right? So basically this tells me, never mind now the details of why we have a sign, a sign here, but basically this tells us that we're trying, the plant is trying to minimize its angle with respect to the angle of the stimulus. It wants to turn its head towards the stimulus. The last term here on the right is proprioception. So this is, you can think of it as a restoring force. Basically, the plant doesn't want to be overbent. This is not a mechanical property. It's an active biological response, okay? And that's it. It turns out that this relatively simple equation is universal enough and can describe these very, very different species and scales. And this is very useful for us because it means that we have a system that we understand both from the experimental point of view of plant tropisms in response to light and gravity and so on, but also from the mathematical framework point of view. So now going back to the question at hand, I was discussing structures, right? And the emergence of these structures, complex structures made out of many individuals. So to understand where these structure comes from, it might be a good idea to look at the dynamics. So here there's a beautiful movie of the zebrafish nervous system development. And what you see here is basically the single neurons growing together to form the structure of the nervous system. And I think that to many of you, this might feel somewhat reminiscent of the very well-known movies of collective dynamics and self-propelled systems, right? Like the movie here on the right, where you see a group of fish moving in an orchestrated way. So maybe there's something similar, there are similar components between the orchestrated growth dynamics that we see in, for example, this nervous system, but also in root systems and so on, similar to the components that underlie these well-known collective behavior systems of motile systems. So the idea is the following: we know that for collective behavior, there are many models, of course, but they all boil down to similar ingredients. So we have many individuals, which is what you see here, the arrows at the top. And now let's consider the arrow in red. This is one specific individual, and it interacts with all other individuals within a specific radius of interaction, okay? So this is the blue circle. And generally you have long-range attraction and short-range repulsion, and that is and noise, of course. And this is pretty much enough. These knobs of changing the interaction range or the amount of noise, this is enough to get the variety of types of collective dynamics or phases, if you will, that we see in nature. So you see here at the bottom, you have a random movement. You have the torus's milling movement of fish, for example, and polarized movement, where you see many times in fish and birds where they all move in the same direction. So this is a kind of the building blocks of collective behavior models. So now if we think of what would the analog model look for growing systems. So now each individual is not motile, but growth driven. So these are these arrows. And you can imagine that if we look at the red arrow, okay, this is our one organ, it will interact similarly with attraction and repulsion to all other growing organs which are within its radius of interaction. But then the phase space, let's call it, or the state space, would be a structure, right? So we can imagine, these are drawings as you see that I made, so these are completely a figment of my imagination right now, but I suggest that the analog of the random movement of motile system would be a random network. The milling movement would be, let's say, a helix, and the polarized movement could be a bundle of co-moving or co-growing organs. So in any case, the phase space would be inherently different, because now we're talking about structures or something. In any case, you have memory, since if you have a system that's growing and you continuously change your environment. Okay, so everything that we know about collective behavior from motile systems basically can chuck out the window, and we have to rethink everything and redefine everything. And this is what I'm going to tackle here. I'm going to basically show here the beginning. I'm going to lay down the foundations, let's say, for a mathematical framework which will allow us to ask these questions. So if we focus at this one organ interacting with other organs, right, we can assume for a moment, for simplicity, that each organ can emit a signal from its tip. Okay, and this is what all organs sense as well. So now my one red organ is sensing basically one point. This is a bit different from the models that we have for tropisms, because in tropisms, the stimulus is assumed to be at infinity, like light or gravity. And then we have a vector field, and the whole organ basically senses the same direction of the stimulus. Here, since it's close by, different points on the organ sense a different direction. So we need to account for this. And basically, this is our generalized model, which is based on the mathematical model for regular tropism. But now we have an angle of the stimulus with theta p, which it can change. It's also a function of s. It's not the same, right? It depends on where you are on the organ. And also you can imagine that the lambda, which is here represents the interaction radius, right, or the sensitivity, also depends on your distance from the point, because we want it, later on we will want it to depend on the distance. But the idea is the same concept as before for regular tropism. Now we can also define various characteristic scales. So we have two length scales. One is the convergence length, LC. The convergence length basically tells me on which part of how big is the area which will be curved as a response to some stimulus, okay? You can think of it as the radius of the maximal curvature that the plant can reach. Another length scale is the LGZ, or the length of the growth zone. So in plants, not everything can grow all the time. There usually is a finite growth zone, which is the one that only if you are within the growth zone you can respond to a stimulus. But as you grow older at the bottom of the plant, sorry, you will see that basically the plant does not respond anymore because it's not growing anymore. So the response can only occur within the growth zone. These are the two length scales. There are also two time scales. There's a convergence time. This tells me what is the time until I reach my steady state form, okay? And there's the arrival time, or first arrival time. And this is the time in which I reach the direction of the stimulus for the first time. So you can imagine that if I overshoot this, okay, and then I converge, the arrival time will be shorter than the convergence time, but it could be also the other way around. Now, if I take the ratio between the two time scales or the two length scales, it turns out that the relation is exactly the same. The two ratios are exactly the same, and they give me a dimensionless number, okay, which we call here Z, and it represents basically some sort of equilibrium, if you will, between sensitivity and proprioception. So this is between my sensitivity to some signal, how much I want to respond to it, and proprioception, how much I want to, how much restoring force I have, or how much friction, basically, I have in this. To understand this, to give a bit of a feeling as to what this Z means, let's look on the right-hand side. There is a low value of Z. So here it means that the convergence time, for example, is much smaller than the arrival time. I converge before I arrive to the tip, as you can see here. Okay, so basically I never reach the direction of the stimulus. On the left-hand side, you see a high value of Z, and here I converge much after the time of arrival. And so you see that you had these oscillations, okay? Oscillations and overshoots. This will be important later. And I'll just show you that these represent, for example, these two different types of movements. In wheat, you reach the steady state. There are no oscillations or overshoots, but in Arabidopsis inflorescence, there are oscillations and overshoots. Okay, very quickly, because I'm not sure how much time I have with all the technical issues I've had. So one thing that we need in order to understand what happens with interacting organs is to understand what are the possible positions of the tip. So what you see here is basically, for example, on the right, you have for Z equals 10. At the x-axis and y-axis are just the x and y positions. So at 0 and 0, where you have the red cross, that's the base of our organ. And now I basically, what I do is I put point signals, point stimuli at different points in space. For example, the red dot that's outside of the box here. And then I check numerically, of course, where the tip, when my organ reaches steady state, I check where the position of the tip is. And I remember that. Then I scan for all of the possible tip positions. Okay, so this is another example. I put a stimulus at a different point, and now I check where the tip is. And as you can see here, they all fall, at least from on this side, on the same line. Okay, there's a line that's common to all distances. So the different colors represent how far away the point is from my organ. Okay, so yellow is infinity, for example. That's regular tropism. The point is that there is the outer part, this outer trajectory represents all possible points for a tip with a given Z. For a lower value of Z, as on the left, which is stiffer, right? It doesn't like to be bent. You'll see that it doesn't reach as far down. It doesn't like bending on itself. So now let's go let's try to model this interaction between pairwise interaction, two organs, okay? So I have organ 1 on the left and organ 2 on the right, and their distance, they're symmetric around our y-axis, and the distance between the bases is delta x, and the distance between the bases along the y-axis is delta y. And what we do here is that we say that they both emit signals from the tip. So these are the red dots, okay? And one organ can sense the signal from the tip of the other organ and each other. So now the angle of the stimulus is this theta 1, 2p or 2, 1p. This is the relative angle between the two tips, okay, as you can see in the drawing here on the right. And now we have two coupled equations, one dynamical equation for each organ. So organ 1 is the first equation, so we have kappa 1 and theta 1, and the same equation for kappa 2, the curvature of the second organ. And basically, they're coupled through this angle theta 1, 2 or theta 2, 1, which is the relative angle of the tips. Okay, so we have the same equation from before, point tropism, but now the point can move. The stimulus is not fixed in space, but it can move because it's also on the other organ. Okay, so here you can see the coupling. So how do the dynamics look like? So on the left, I show a sort of a state space, if you will, and I'll explain a bit how to read this. So the x-axis and y-axis are delta x and delta y, the distances between the bases of the organs, and the left plot, the color tells is the value. The color tells me, the color code tells me what is the value of the x position of the tip of the organ number 1, okay? And on the right, the picture is the Y position of organ 1, the one on the left. So just to make sure that it's that we can understand this, basically, like before, yeah, the left organ is always, the base of the left organ is always based at 0, 0. Okay, so as you can see here, it's the red cross on the left. That's basically the base of the left organ. And now when I look at a specific pixel, okay, for example, the one which is the second red cross, that represents that in that position, what would happen if I would, what would happen if at that position I would have placed the second organ, organ number 2, the one on the right, I would place the base at that position, okay? But then I ask, basically the color tells me what is the X or Y position of the tip of the left organ. This is a bit complicated to grasp very quickly over this talk, which is I'm a bit rushing through this, I'm sorry. But the point is basically that let's take first of all the center image here. So where I placed the right cross, the right organ with a red cross, since we see the value of the pixel is green, okay, it's a positive number between 0 and L. This tells me that the position of the tip of the left organ is positive. It's above the x-axis, pretty much as you can see here in the figure that I showed here. Now, if I go to the left panel, where I look at the value of x position, right? Now I see that in the same spot you have a pink value. So this means that you have a negative value for x1. So what this tells me is that I am to the left of my y-axis, which is again exactly what I drew here with the 2 organs. Okay. So let's try, now that we roughly understood what this means, let's try to look at specific examples. If I look here at position, I put there are numbers on this state space, 1, 2, 3 at the center, and then 4, 5, 6, and so on. So these are different, let's look at 2 examples that are with delta y equals 0. Okay, so I'm only looking at, they're symmetric, completely symmetric, and I'm only looking at different distances delta x. So at position number 2, which represents delta x equals 1.2, basically what we see from the color, this is the final state, okay? The two, and you see that they touch at the tip. Now the 2 circles that you see at in the background, they represent, if you remember earlier on, I showed you all the possible positions of the tip. So that's what they represent. And what we see here is basically the steady state is where the tips touch at the first intersection point of these 2 circles. Now, if I look at the next state, which is state number 3, marked on the phase space, this is basically delta x equals 1.3. And now you see, if you look at the colors of the y values, right, it went from green to pink, from positive numbers to negative numbers. So now the tip is below the x-axis. And if we look at what this means what this means relative to the possible apex positions, this means that they're touching down at the second intersection. So if you can see that there are these very strong discontinuities in the type of states, like being up or being down, which means the green part or the pink part in this phase space or state space. And this tells us that the different positions of whether the steady state is up or down is dictated by the distance between them. And what we see is, if we look at the dynamics, so here I'm showing you the movie, this whole simulation until you reach the steady state. And another one for the other one is that the distance tells us whether when the 2 organs reach each other for the first time, will they be crossing each other this way, so going upwards, and this means that they pull each other up, or at a larger distance, by the time they reach each other, they're crossing downwards, and then they pull each other down to the second intersection point. A third type of behavior is basically everything. A black pixel means that you have you have non-convergent dynamics. These two organs will continue to oscillate forever, okay? And this is due to the fact that they're always curved in a way that the base senses one direction, but the apex senses a different direction. And by the time that the base tries to compensate for this, the apex already goes in the opposite direction. And this basically gives us these non-convergent dynamics. So I think my time is pretty much up here. I'll just say then, wrap this up and say
Hi, Yasmine.
where this is headed to, right? So, yeah.
Just to jump in, you can take another minute. That's totally fine.
Okay, super, thanks. Because I put a timer here and it started beeping and I got all, yes.
No, you're good.
Thank you. Basically what I showed here before is a rather, we only discussed pairwise interaction between two organs, but notice that the organs do not grow here, right? I didn't mention this, but we're disregarding growth here, and we're just taking it as the implicit driver of the movement. But of course, this is far away from the type of movies that I showed at the beginning that gave us the motivation for this whole problem. So, but the point is that basically what we have here now is a framework which allows us on which we can build basically our model. So what we will want to do now is to add, first of all, growth, and that's relatively simple to do. You just need to add a material derivative. Look at 3D, which is a pain, but it's doable. And of course, elasticity and so on. And then basically the only thing that we will have to do later on is to generalize, instead of just looking at pairwise interaction, put many organs. So in terms of growth in 3D, we're able to do this now. So this is unpublished, but hopefully submitted within a week. So for the best. What you see here is an organ growing in 3D. The black part is outside of the growth zone, so it cannot change its shape anymore. The green part is within the growth zone, so that is what responds to stimuli. You have, in the center video, you have a small blue dot, that's a point stimulus. So you see how the whole organ grows towards it. On the right, we have a rod stimulus. So we get basically twining of climbing plants, for example, for free, even though it's cheating, because this isn't exactly real twining. But still, this just shows you the kind of the rich type of dynamics that we can get with this. So now basically all that's left to do is to put these two things together, the pairwise interactions with this 3D growth, have multiple interactions, and then we'll see what kind of a phase space we get. And hopefully we'll be able to look at what we want to do at the end is to take a real system, whether it is a neuronal structure or root structure or fungal structure or whatever, extract the dynamical characteristics of the single individual, plug it into our model, and then predict the type of structures that we can get. So if anyone here has a system in mind that might make sense to use with our model, we'd be very, very happy to collaborate, of course. And that's it. So thank you very much for organizing this again and for listening with all the technical issues. I hope I managed to come through after all, and I'll be very happy to answer any questions you may have.
Yay, thank you.
Thank you.
Thank you for leading us off into this wild unknown for us too. So it was a wonderful talk. We're going to try the question session, and we haven't done this before yet, so we're going to get a sense of how this works.
Okay.
So there's a couple, before we get into the questions, I just wanted to say that we enabled the chat feature to be visible to everyone. So everyone can now see the chats that you write. And I guess what we'll do is, if you have a question, raise your hand, and we'll go through and call you out in order. Okay? So let's try someone raise their hand, and we'll see what we can see here. Oh.
Who wants to be technologically brave here?
Someone. Oh, there's a hand.
Yeah.
Okay.
Nikhil?
Nikhil, go ahead and unmute and ask your question.
You're actually already unmuted.
No? Can everybody hear? Did we lose all?
No. I think Yasmine, are you there?
Yep, I'm here.
Okay,
Okay.
Hear me?
let's try another question. From
Just pick. Yeah.
from Hang Tang.
Hey, can you hear me?
Yes.
Yes.
Oh, perfect. Yasmine, I actually have a question for you. So you were talking about the comparison between your model and the bird flocks and fish flocks. But something I noticed that might be a little bit different is that I think you have a restorative force or restorative term, which kind of tends to flatten out the trajectory and make it straight. Is that the term that you may have to get rid of if you want to consider maybe a bird trajectory where the bird has already flown through those paths and cannot basically correct those past trajectories anymore?
So I hope I understood your question correctly. I should just say that this restoring force that you said that it makes the trajectory straight, that's
Right.
true for the constant stimuli of light or gravity, that they don't change in time. So once you straighten out, that's it. You're stuck in this trajectory forever. But for a point stimulus, particularly if you look at, for example, the 3D movie that I showed at the end, basically you continue to grow until you reach your point. But if the other point moves as well, then you would basically follow it in the environment wherever it goes. So in that sense, you're not constrained in terms of your dynamics. Where you are constrained is that once you form a trajectory in space, you can never go through that space again because it's taken, right? So that's I guess the most critical difference, that you continuously fill up space and therefore you change your environment as you grow.
Got you.
I'm not sure that answered your question.
Yeah, that's perfect. Thank you.
Sure.
Yasmine, I have a quick question.
Sure.
In terms of the interactions between organs, so branches in this case, would you expect something like excluded volume or also some attraction? Is there any reason to think that branches would be attracted to each other?
You mean branches in trees? You're talking about the specific system or any type of these systems?
I was thinking about plants in this case.
So that's a tricky question. Branches, I'm not sure. My candidate here for a system was actually root systems, because in root systems we know that there are interactions because basically each root has a very distinct signal, whether it's root exudates, so it means that you're constantly having chemicals released from the root tip, or just through the gradients of nutrients and water uptake around you. And we know that plants respond to these sort of gradients. So there are interactions, but they're not well observed. Okay, they're just observational, and there's very little quantitative work on this. So, for example, one example is when you have roots growing on agar, there's also the issue that you can't see root growth when it's in ground. So that's actually a very big issue with these studies. But people sometimes study roots growing on hydrogels, and then you see that they tend to, if you look at pairs of roots, specifically Arabidopsis, they tend to grow one towards each other. So they could, it's not exactly obvious what is the nature of this attraction. It might just be surface tension and not anything related to chemical attraction, but it's there. And in any case, so I guess it's more, it sounds reasonable that in plant roots at least, and probably also shoots, right? You should have more repulsion because the goal is different. I mean, the goal there is to forage as much as possible and not to get into each other's, you don't want knots, right? You don't want to grow into a fellow root. So you want to grow as far away from each other. So there I don't expect actually to have attraction as in flocks, because we don't want adhesion or cohesion, we want the opposite.
Okay, thank you.
I think that was, so thanks again, Yasmine. I think we're going to move on to keep sort of vaguely close to the schedule, but thank you so much for your interesting talk and for opening up our session.
Thank you. And I'm sorry that I don't think I'll be able to,
There's more questions.
just because